USP Apparatus 4 for dissolution testing is also called as flow-through cell apparatus. This apparatus is not used widely as the apparatus is specialized for some of the formulations type. USP Apparatus 4 is officially recognized for dissolution testing in the USP. United States Pharmacopoeia provides information on the flow-through cell apparatus, that is USP type 4 apparatus for dissolution testing. As the name indicates, flow-through cell, it is consisting of the flow-through cells made up of glass or stainless steel sometimes. It is having a piston pump or peristaltic pump. It has media reservoir and a filter system and sample collection system. The formulation or dosage form is placed inside the cell and the dissolution media continuously flows through it at a specified or controlled flow rate. That's why the name is given as flow through cell apparatus. What is the principle for the operation of this apparatus? It operates on open loop or closed loop system. In open loop system, fresh medium continuously flows through the cell and collected for the analysis. Closed loop medium is recirculated, allowing drug concentration to be monitored over time. Laminar flow mimics in vivo conditions better than the static systems. So it is one of the advantage. Key operating principles are flow rate, temperature, cell types, sample volume, and the media volume. So if you say that the media is moving through the cells, that's why the media volume can be flexible. As you can see here, there is a media reservoir, there is a pump to force the media or dissolution media or composition through the pump. It goes to the flow cell and then it comes in contact with the formulation. Then it goes to the fraction collector. The parameters or operating parameters are flow rate, which may be from 4 to 32 ml per minute, but this apparatus is typically operated at 8 to 16 ml per minute. Temperature is 37 degree. Cell types are small volume standard and the large volume. Sample volume is variable based on the formulation and media volume is flexible. You can see here the flow through cells made up of glass. Then what are the specific applications? Why this type of apparatus is designed? So it is mainly used for the modified release formulations like ERDR, poorly soluble drugs, transdermal systems, implants, suspensions, soft gelatin capsules, beads, microspheres, and it is also suitable for the biorelevant dissolution testing. Main advantage or application is for using it for the formulations containing the poorly soluble drugs. Advantages of the flow-through cell apparatus are continuous flow mimics physiological conditions. It is suitable for low solubility drugs where sink condition is critical as the media is moving or it is flowing. Then media can be changed dynamically to simulate pH progression like stomach to intestine. And it is ideal for the controlled release profiles and it works with small and large dose ranges. These are the advantages of flow through cell apparatus. Limitations for this apparatus: it is more complex compared to USP 1 or 2 or USP 3. It requires precise flow control and it is not ideal for immediate release type of formulations. If the formulation is IR type, then directly we can use basket apparatus or paddle apparatus. It has higher operational and validation efforts because it is more complex than the apparatus 1 and 2 and 3. Why to use USP apparatus 4? So when standard apparatus like basket and paddle apparatus cannot give you the results or are inadequate to replicate the in vivo environment, when biorelevant media transitions like gastric to intestinal pH are needed for some of the complex formulations, at time USP4 can work. For early phase development where flexibility in flow rate and medium volume is essential. Then what is the regulatory and industry acceptance for this apparatus? So USP and EMA endorse apparatus 4 for specific formulations and dosage forms. It is accepted in IVIVC studies and it is frequently used in the biopharmaceutics classification system studies. In conclusion, USP apparatus 4 is flexible and robust tool for resolution testing, especially for the complex formulations. For some of the complex design, modified release, controlled release formulations, some of the soft gel formulations, and also it is more useful for the poorly soluble drugs. Also, some formulations are there which are not disintegrating in nature. For those, this apparatus is suitable. It enables better simulations of physiological conditions, improving the prediction of in vivo drug behavior. So for these reasons, the USP4 apparatus is suitable and can be used. See, depending on to the formulation, depending on to the in vivo, in vitro correlation requirements, different apparatus can be used. So USP4 apparatus, it has some merits, advantages for some complex formulations. I hope this video will help you to understand USP4 apparatus. I request you to read more and understand about the USP4 apparatus, because many of the times this apparatus is not used because it is complex and it is required for the dissolution testing of some specialized and complex formulations. Thank you for watching the video and let's meet soon with another informative video.